ஒவ்வொருத்தரா வராங்க.
Banu ma'am. Banu ma'am. Subramanian sir.
Yeah, yes sir.
Good afternoon sir.
Good afternoon sir.
Banu ma'am இருக்காங்கல்ல?
Jain sir, good afternoon.
Hello. Banu ma'am.
Aaron, good afternoon.
Good afternoon ma'am. Good afternoon.
Good afternoon.
Madam நீங்க பேசுறது
Good afternoon.
மேடம்.
ஹலோ.
நீங்க பேசுறது பிரேக் ஆனிச்சு. நான் பேசுறது கிளியரா கேக்கா?
இப்போ கரெக்ட். கிளியரா இருக்கு.
சரி மேம். சரி.
ஓகே. Hello. Nityanandam sir.
Yes, madam.
Can you wait for 4 minutes, sir?
Yes, madam. No problem. No problem. No problem.
Because this is untimed, no. That's why.
No, I understand. It's okay. It's okay. No problem.
They're taking time. Okay.
No issues. No
Please,
issues.
sorry. Okay.
Yeah.
Saravanan sir. Hello.
மேடம் சொல்லுங்க மேடம்.
ஸ்டார்ட் பண்ணிடலாம் சார்.
ஓகே மேடம்.
Ma'am, shall we start, ma'am?
Yeah, Saravanan sir will put the song.
Okay.
He'll share the college song.
வாழ்க்கையெல்லாம் கல்விதானே வரமாகும். காமராஜ் கல்லூரியில் பொன் விழா கோலம். பொன் விழா பொன் விழா பொன் விழா பொன் விழா ஆண்டிலே ஆனந்தம் கொண்டாடும் நேரமே இது காமராஜ் கல்லூரியில் பெருவிழா சந்தோஷ பெருவிழா. இது காமராஜ் கல்லூரியில் பெருவிழா சந்தோஷ பெருவிழா. இடமே மதித்திட கல்வியில் உயர்ந்து நின்றோ. கல்லூரியே
சோலைவனமானதே
பூக்கள் சூழ
பெண்கள் வந்து வீசுதே.
கல்லூரியே
சோலைவனம் மனதே
பூக்கள் சூழ
திங்கள் வந்து வீசதே வாழ்க நம் கல்லூரியே பாடு மாணவனே
வாழ நம் கல்லூரியே புகழ் பாடு மாணவியே வாழ்க்கையெல்லாம் கல்விதானே வரமாகும்
யா யா யா
காமராஜ் கல்லூரியில் பொன் விழா கோலம்
யா யா யா
பொன் விழா
பொன் விழா
ஓஹோ பொன் விழா
பொன் விழா
அன்பின் வாரில் காமராஜா ஏழைகள் மரபில் ரோஜா அன்பின் வாரில் காமராஜா ஏழைகள் மரபில் ரோஜா
காமராஜர் நல்வழியை நாம் தொடர்ந்திடுவோம்
காலம் போற்றும் கர்ம வீரரை புகழ்ந்திடுவோம் ஏழைகள் வாழ
ஏழியாய் இருந்தால்
எங்கள் வாழ்வில்
கல்வியை கொடுத்தார்
ஏழைகள் வாழ
ஏழியாய் இருந்தால்
எங்கள் வாழ்வில்
கல்வியை கொடுத்தார்
யாக வாழ்க்கையற்றவர் நம் வாழ்வில் ஏற்றம் தந்தவர்
யாக வாழ்வையேற்றவர் நம் வாழ்வில் ஏற்றம் தந்தவர்
வாழ்க்கையெல்லாம் கல்விதானே வரமாகும்
யா யா யா
காமராஜ் கல்லூரியில் பொன் விழா கோலம்
யா யா யா
பொன் விழா
பொன் விழா
ஓஹோ பொன் விழா
பொன் விழா
பொன் விழா பொன் விழா ஆனந்தம் கொண்டாடும் நேரம் இது காமராஜ் கல்லூரியில்
பெருவிழா சந்தோஷ பெருவிழா
இது காமராஜ் கல்லூரியில்
பெருவிழா சந்தோஷ பெருவிழா
Thank you, Saravanan sir. Start பண்ணவா? Hello,
Ma'am.
Saravanan sir. Okay. No water, no life. No blue, no green. Good afternoon, everyone. I am Dr.K.BHANUMATHI, Head, Department of Zoology, Al-Ahmad College. I welcome you all for this webinar. The Department of Zoology, Kamaraj College, proudly organizing this webinar topic on habitat enhancement efforts in Kuwait, Arabian Gulf. First of all, I would like to thank you all for taking time out and being here today, circuit breaker time. Before starting the webinar, I would like to share a few instructions. Please, all the participants, kindly mute your mic and the video throughout the session. If you have any questions and if you want to communicate with us, I mostly welcome, and please share your questions in the chat box. We will send you the feedback link in the chat box after the session, which will be active for next 20 minutes after being displayed in the chat box. You will get the e-certificate once you fill up the feedback form. let's start the webinar in a formal way. I would like to call upon Dr. R. Nagarajan to give us welcome address. R. Nagarajan,
Yes,
are
ma'am.
you ready?
Yes,
Yeah,
ma'am.
you can proceed. Yeah, proceed.
Good afternoon to you all. I would like to welcome one and all present to this webinar about habitat enhancement effort in Kuwait at Arabian Gulf. I especially want to welcome our college principal, Dr. D. Nagarajan, Secretary, Mr. A. Rajendran, IQAC Coordinator, Dr. J. Pungudi, Head of the Department, Dr. K.BHANUMAThi, other faculties members of our department, Dr. J. Nagarajan, Mr. Subramanian, other staff members and students of our college and staff from other colleges. Welcome to all. My special greetings to our chief guest, Mr. M. Nithyanandan, Assistant Research Scientist, Kuwait Institute for Scientific Research. Welcome, sir. I hope all will enjoy this webinar. Thank you.
Can you hear me?
Yes.
Okay. Thank you, Dr. Aran, for your welcome address. Next, I request our respected principal, Dr. D. Nagarajan, to deliver his felicitation. Sir, please.
Good afternoon, Dr. Bhanumathi, the head of the Department of Zoology, Kamaraj College, and the convener of this international webinar on habitat enhancement effort in Kuwait, Arabian Gulf, faculty members of Zoology Department, the resource person Thiru Nithyanandan, Assistant Research Scientist, Kuwait Institute of Scientific Research, dear students and participants from other colleges, good afternoon to everyone. It gives us immense pleasure to have Thiru Nithyanandan from Kuwait today as our resource person. On behalf of all the stakeholders of Kamaraj College, I warmly welcome our resource person. Arabian Gulf is also called as Persian Gulf. Persian Gulf lies between the Arabian Peninsula and southwestern Iran. The Gulf is shallow and rarely deeper than 90 meters. The Arabian Gulf is characterized by extreme environmental conditions, that is extreme temperature with high salinity and turbidity. In Kuwait, the increasing population also increases the pressure on fish stocks and are nearly overexploited. These overfishing activities declined the health and sustainability of Arabian Gulf ecosystem. Now, Kuwait imports fishes to meet its demand from other countries. Coral reefs are the living museums of the seas. They are also called as rainforest of the sea. They are the home for most of the marine animals, including fishes. In the earth, coral reef occupy about 1% of the earth's surface, but they give shelter for 25% of the marine species. Hence, coral reefs are very important in terms of biodiversity. The coral reef fishery is important for supporting the economy of coastal populations. Coral reefs also give protection to the seashore from the wave actions. In recent times, the coral reefs in Arabian Gulf have been exhibiting bleaching and degradation. The main causes are the rise in temperature, ocean acidification, deposition of direct and indirect human waste into the sea, discharge of pollutants, especially oil and other effluents, into the seawater. These various activities impact the health of coral reef and also the symbionts zoos and the lake. In Arabian Gulf, the temperature fluctuations between winter and summer is about 20 degrees Celsius, whereas in other regions, the variations are only below 5 degrees Celsius. Due to high temperature fluctuations, only 10% of the coral species are available in Arabian Gulf when compared to the reef in other coastal regions of the world. During 2016, more than 70% of the corals have been affected in Kuwait seawater. Reef restoration initiatives have been undertaken to prevent further damages to seabed and reefs. Installing the buoys around the critical areas and installing artificial reef balls on the seabeds to foster coral growth are going on. So effective management of fish resources and also the restoration of coral reef are necessary to save the coral habitat in the Arabian Gulf. I request the participants to make use of this very important webinar. You can understand the various measures being carried out in the habitat enhancement of Arabian Gulf. At this juncture, I thank our resource person, Thiru Nithyanandan, for accepting our invitation. I sincerely appreciate the convener, Dr. Bhanumathi, and other faculty members of the department for organizing this webinar successfully. My warm felicitations and greetings to all the participants. Thank you.
I thank our principal for his valuable felicitation. Thank you, sir. Let me invite Dr. J. Nagarajan to introduce the speaker, Mr. Nithyanandan. Jain? Mic mute la irukku.
Good afternoon, madam.
Good afternoon.
Good afternoon, everyone. Mr. M. Nithyanandan graduated from Madhavi Christian College, University of Madras, with a bachelor's degree in zoology, specialization in industrial fission fisheries, and then completed a master's degree in oceanography and coastal area studies from Al-Ahmad University in Kavikudi. He started his research career in Zoological Survey of India, Chennai, during 2001 to 2003 as a junior research fellow. He was involved in studying the reef-associated echinoderms of Gulf of Mannar. In 2006, joined the Mega Coastal Development Project, Sabah Al-Ahmad CCT in Kuwait to manage its environmental development. He monitored the CCT for the next decade to study the benthic colonization in man-made marine habitats and conducted habitat enhancement and restoration experiments. He also is had a certified open water diver with diving experience in India and Kuwait. Recently, he joined as an assistant research scientist in the oceanography, a group of Kuwait Institute for Scientific Research. His research area include biodiversity, benthic ecology, and habitat restoration. Has 19 years of research experience and has several publications in wide international journals to his credit. Recently, a new species of host shrimp collected from Kuwait was named after him, K.BHANUMATHI Nithyanandam. Sir, thank you, sir. Welcome. Welcome. Thank you.
Thank you, Dr. Jain, for introducing our chief guest today. I request our speaker, Mr. Nityanandan, to proceed the session. Sir, please, you can share the slides and you can start.
Thank you, madam. Thank you.
Thank you. Welcome, Mr.
Yeah, thank you. A very good afternoon to all of you. First, I would like to thank the principal of the Kamraj College, Dr. Nagarajan, and the head of the department, Dr. Bhanumathi, and the other staff who had invited me to deliver this lecture for this webinar. It's a great honor for me. Thank you so much. Without taking much of time, I would like to begin with the webinar, and because I have quite a lot to show and discuss with you people. So the topic of this webinar for today is habitat enhancement efforts in Kuwait, Arabian Gulf. So I'm going to talk about 3 different habitats, basically mangroves, seagrass, and coral reefs. How these habitats were established in a man-made marine habitat in Kuwait. So this is Arabian Gulf. It's a very shallow sea, a marginal sea, which is around an average depth of 30 meters, and the salinity is very high here, like around ranging from 42 to 65 parts per thousand from north to south, if you see. And also the atmospheric temperature drops to 5 degrees in the winter and shoots up to 50 degrees in summer. And the water temperature range is around 10 to 33 degrees. Gulf is a very fragile coastline, and also it is a very extreme environment. But due to the oil wealth and due to very high income due to oil resources, these countries have ventured for a lot of coastal development activities along the coastline. Different countries have their own projects, and this is to attract the tourism in the Gulf countries. So one such project, the first project in Bahrain is Durat al-Bahrain. This is the one. This is basically a group of artificial islands constructed by dredging the sea. And also it contains a series of artificial lagoons as well. Followed by Bahrain is the United Arab Emirates, the famous Palm Island. This is another interesting coastal development project, a very successful real estate project also, and a structure which is even visible from the space. Palm Island has attracted a lot of media, and also it is one of the unique development projects in the Gulf, and it is a massive project. There are about 3 different projects, but again, one of the projects is still yet to be completed. If you see this Palm Island, and in the Palm Island, there are a lot of leaf-like shapes, and all these have a beach and the villa or apartment. So that is the concept basically. Each house should have a beach. This is a very massive development and very successful in the Gulf. And the third one is from Qatar, is the Qatar Pearls. This is the shape of a pearl. So all these developmental projects in the Gulf are pretty popular, and also they are equally criticized from the environmental point of view, because there is some level of intervention in the environment, because all these projects have been built by dredging the sea and also backfilling it. So all these projects had their own setbacks. But again, from the environmental side, although there are some developments, but again, there's a lot of criticism. But apart from these projects, Kuwait had a different kind of an ideology. Kuwait, if you see, is a small country in the northwestern Arabian Gulf. It is around 17,000 plus kilometer square area with a coastline of less than 500 kilometers square. Sorry, 500 kilometers. And it has a population of around 4.137 million. And Kuwait is a very rich country, one of the richest country in the Gulf due to its very high oil resources. And following the other Gulf countries, Kuwait also wanted to have a similar coastal development project, on a slightly different ideology, apart from what has been happening so far in the Gulf. Moving on. So this is Kuwait again, and on the southern shores of Kuwait, there is a place called Al-Khiran, where a huge coastal development project was planned. So this project was a different concept, because apart from other projects in the Gulf where they are venturing into the sea, the development, the company which handled this development, they wanted to dig the land and bring the sea into the land, basically bring the sea into the desert. So this was a totally different concept actually, and there was less intervention as far as the environment is concerned. So this is the largest coastal development in Kuwait, and it is supposed to be completed in 2020. It is a phased development with 5 phases. The last phase is under construction, covering a total area of around 70 square kilometers, equal to the size of the city of Manhattan in USA. So in this project, there's a lot of interesting things happen, which I'm going to share with you now. So actually, if you see, there are 2 creeks in this area. These 2 creeks here. This is a red arrow mark. These 2 creeks were modified into a series of man-made lagoons by excavating the land. So this is how it is now. So these finger-like lagoons are man-made lagoons. All these lagoons are oriented towards the direction of the wind, and the exchange of water is through tidal forces only. There is no artificial circulation. So this is totally a very different concept. And also they brought the sea into the land by around 9 kilometers. So this is how it is, this aerial view of the Sabah Sea city. On average, the depth will vary from 3 to 9 meters depending upon the location. In some areas are deeper, some areas are shallower, because it has to fit the hydrodynamic design and the model. And they have done extensive modeling to see whether the flushing is appropriate, whether there is any proper circulation issues, or any issues with the water quality, or are there any dead points where there is issues with oxygen, things like that. All those physical chemical parameters have been looked into, and they did a very detailed modeling, and it was very successful. Till date, there is no serious water quality issues. So in this project, we were asked to do different type of habitat enhancement projects. So before going into that, I would like to give you a small introduction about the habitat connectivity in tropical ecosystems. In reality, if you see, this is how it is in a tropical ecosystem. There's terrestrial vegetation.
Sir, excuse me. Sir, excuse me.
Yes.
Sir,
Yes.
Nityanandan, sir. Sorry to disturb you.
Yes.
Somebody is annotating. You please share the slide again. Stop it and share the slide again.
1 minute.
Students, please, ulla irundhu yarum edhum pannakudadu. You will be severely punished. Please don't do anything.
I could see that, but I'm unable to do anything from here.
Screen stop pannittu.
1 minute. I'll do that.
எல்லாரோட கண்ட்ரோலும் எங்க கிட்ட தான் இருக்கு. If I find out, you will be severely punished. Please don't do that. Yeah, thank you, sir.
Okay.
Sorry for the inconvenience. You can start.
It's okay, madam. No problem. It happens. It's okay. See, this is the schematic diagram showing the connectivity in tropical ecosystems. So this is how, in nature, the zonation happens. Like there is terrestrial vegetation followed by mangroves, and then seagrass and coral reefs. There is a strong connectivity between these habitats. So all these habitats, they're how valuable they are. For example, mangroves, they bind sediments. They're very helpful in nutrient cycling and organic material export, and they also serve as a nursery. All these ecosystem services are well known for all these habitats, even for seagrass. So there is a strong connectivity between these habitats. So you can see this arrow marks indicating it. So we were thinking in these lines basically. So if you wanted to enhance the value of the habitat, so we need to establish this kind of habitat so that there will be some level of connectivity in this artificial habitats what we have created in Gulf or in Kuwait. So this is the habitat enhancement project planned for the sea city. First, we had a question whether it will be possible to establish this habitat connectivity, what we are really seeing in nature. Initially, the Kuwait government, there is an environmental agency by name Environmental Protection Authority, EPA. They suggested us to transplant mangroves first as a first step. So that was planned for these 4 islands here. These are small artificial islands. In these islands, we have to transplant the mangroves. We started this project in 2006. And during the course of the project, as the project started developing into different phases, in 2009 onwards, we started seeing seagrass colonization. This is natural colonization without any transportation efforts. And followed by this, we saw coral colonization also here. These are hard coral, basically non-branching corals, which were encrusting and growing on rocky structures. And seeing all this happening, and also the bamboo project was ongoing, then we thought we should enhance the project with different habitat enhancement efforts. Like we wanted to establish a coral garden in these 2 sites, which are nothing but a rock island. These are some of the projects that we had planned. So then we started moving forward. To begin with, we started the mangroves. This is the Mangrove Establishment project in 2016 to 2015, almost 10 years. This was a very challenging task for us because the Kuwait's environment is very harsh for mangroves. And the temperature dropping around 2 degrees in the winter, and in summer it shoots up to 50 degrees, and water temperature also is too cold for mangroves. And apart from all these things, we have frequent dust storms and also hailstorms during spring. So all these natural events kind of pose a lot of concerns for us to establish mangroves. So the first step in establishing a mangrove is we have to select a site. So that site has to look in terms of the following characteristics in terms of tidal regime, because you have to see the inundation, you have to see the soil conditions, how the wind direction is, and then you have to select a suitable species, a candidate species. For this project, we selected Avicennia marina because it has been very successful all over the world, and also it is locally available, and we were able to import seeds from Abu Dhabi. So there was a local ecotype available. So we went for Avicennia marina. Literature also indicated that other species had failed here in Kuwait. So we selected Avicennia marina, the grey mangrove. So these were the areas where the mangrove transplantation was done in the artificial islands of Sea City. So this is the germination process. When we collect the propagules, we have to soak them for 24 hours in the water and then sow the propagules in the plastic pots with around 70% of desert sand and 30% potting mix with the peat moss. And within 10 to 15 days, they started germinating. After 2 weeks, they were like this, as you see in this picture here. So after the germination is done, you have to slowly harden them before you transplant them into the respective site. So what is hardening? Hardening is initially when you germinate the mangroves, they will be germinated in fresh water. So once the propagules are germinated, then they have to be slowly introduced to the saline water, seawater. So if you just start irrigating with salt water directly, then it will die. So slowly you have to increase the salinity. First stage was 25% salt water with 75% fresh water. And then second stage was 50-50. Third was 75 salt water and 25% fresh water. And the fourth stage was 100% salt water. So each stage is around 15 days. So this happened during February and March. So then, followed by this, we moved them into intertidal nursery so that they should get used to the environment where they are going to be transplanted. So this is just an acclimatization process. This is another 50 days. So after that, we started transplanting them around May to June. So this is how they prepare the holes, and the mangroves seedlings are transplanted at the 1 meter interval between each of them, and in between the rows also slightly more than 1 meter distance was there. So they were transplanted at a level of 0.35 to 0.65 meter above mean sea level. So this level of planting was identified based on the surveys that we conducted earlier. We did some surveys on the slopes at all, so how the inundation cycles are, how the slope of the tidal area. So based on that, we have decided this, and also based on the published literature. So it is not only that just if you plant them and leave them, they will not grow in these harsh conditions. You have to literally take care of them very seriously. So what we did is we have to put cages like this for each and every mangrove. So a mangrove seedling, and also cover them with on top with a plastic sheet and put a rubber band around it. This is to prevent any damage due to dust and hailstorm. Until they grow up to 1 meter height, we have to keep this on. What we do is we keep this covers on until until the early morning, and we remove it in the morning time. Because most of the storm events are in the after, like in the night time or in the evening hours. So what we do is early morning we open this covers and leave it as it is. And before evening we just cover them on top so that there won't be any damage due to storm events. So after this, you have to monitor them. So monthly we recorded the height for at least 100 plants. We tagged them, and each island we measured the height, and we counted the number of branches. We also noted down the foliage condition, whether they are turning brown or they are still remaining green, and how is the foliage development, etc. And also we also looked into the survival and mortality. So how many of them are surviving in each island. So, and above all, we also wanted to monitor the entitled macrobenthos. So we wanted to see the benthic colonization, that how is the ecological association is setting in. So we started looking into the entitled macrobenthos, and yearly we monitored it. And also apart from macrobenthos, we also looked into the birds, mostly migratory birds, which are visiting these places on a monthly basis. So the results, we had a tropical germination rate of 71.8%, and the seeding survival was around 62%. This is above average survival for any other mangrove restoration project elsewhere in the Gulf. Because to achieve this, we struggled a lot. It was almost impossible to grow mangroves here in these conditions, because we have to really train them to get used to these conditions. And if you see here, they won't grow like the normal mangroves what you see in the rest of the parts of the world. All the mangroves what we have transplanted here, they look like bonsais. Mostly they go up to 1 or 1.5 meters height. They don't grow beyond that, but they expand laterally. They become very bushy. This is how the natural population in Bahrain and Saudi Arabia also looks like. But only in Abu Dhabi, it is slightly taller when compared to what we have now here. This is because of the harsh conditions. They have to spend a lot of energy on removing salt from the system because the salinity is very high. So that is why they are not able to spend that energy on the growth. So they are looking very stunted in size. So if you look into the macrobenthos diversity, so that also was pretty high, around 38 species. Sorry. And also, if you look into the abundance, it was close to 4,800 plus. So this is a very high abundance for a transplanted mangrove in terms of macrobenthic colonization when compared to other parts of the world. So, and also birds, migratory birds, we recorded an average of around 100 species between 2006 to 2015 in 10 years period. So we established a population successfully, and also by 2009 they started producing their own tropickles. So we were able to complete the life cycle. What happened is, by 2009 we collected the seeds and we germinated them again in the nursery, and we started raising them. So some of the seeds, they started falling down, and also they started falling down and they started germinating by itself. So this is what you see here. This is what you see here. These two plants which have germinated, and this is all self-germinated mangrove seedlings. So we've completed the life cycle completely, and also they have established a population. We planted around 9,000 meter square area of Avicennia marina in this project, and we were able to harvest around 5,000 to 10,000 tropicals per year. In some years, there was no tropical production depending on the weather conditions. So what lessons we learned from this exercise? So first, we need to identify a proper species, a good candidate species which can thrive in harsh environment like this, and also a careful site selection, which we were able to do it very intensively, to doing a lot of research on different aspects that we have to look into before planting. And then we were able to identify an appropriate level for planting, and then we were able to establish the population. And also looking into the wind direction is very, very important. So we have to construct some sand buns around the island also in order to prevent intensive attack by wind, strong wind, because the dust storms are very intense in the Gulf. So that was a major factor which will definitely impair the growth of the mangroves, and it will eventually kill them also. So we had to look into those aspects as well. And also we have to avoid exposure of waves in exposed shores. Like when they are exposed to strong waves, there'll be strong erosion. So what we have to do is we have to put some plastic PVC pipes wrapped with geotextiles in front of the areas where mangroves have been planted, especially near the fence, so that the erosion can be prevented. So we did all sorts of exercises, and successfully we published this research in 2020, that is this year in June, in Aquatic Botany. Moving on to the next habitat. This is seagrass species, composition and biology and function. So before going into seagrass in detail, what we did with seagrass, we should understand the species composition in the Gulf, and how they reproduce, and what they really have in them, and what are the functions of the seagrass. So as we know, there are about 4 species now in Gulf. Initially there were 3. So the 4th species, Al-Khiran has been identified from Iraq. So there are about 4 species: Al-Ahmad, Al-Khiran ovalis, Triplaceae, and Al-Khiran descipens. So Al-Ahmad is the most common species in Kuwait, and Al-Khiran ovalis also occurs in patches, but because both have different sediment characteristics, Al-Ahmad lives in a more sandy environment, and ovalis loves to live in more sandy, muddy areas, kind of mixed substrate. Al-Ahmad is the most prevalent, most common species in the Gulf from north, in Kuwait, especially from north to south as well. But recently I recorded Al-Khiran Triplaceae, which is the 3rd species to Kuwait. It's a new record. It's an unpublished data set. I'm yet to publish it. If you look into the reproduction of seagrasses here, seagrasses cannot reproduce, as you know, they are flowering plants, as they reproduce elsewhere in the world. They have to really go through the vegetative mode of reproduction. The reason being is the salinity is very high. They cannot invest that energy on reproduction, producing flower and going through seed production. It's very, very difficult. So they go for the vegetative propagation. So what happens is they break down into fragments and they reattach and they grow. This is how they spread in the Gulf. So if you see in the seagrass, especially in Hardening and on the blade, you see a lot of epiphytes, a lot of associated organisms like diatoms and so many other invertebrate groups associated with them. These epiphytes, especially the diatoms and other algae, are kind of preventing them from radiation damage due to the heavy radiation which is seen in the Gulf, because the sunlight is pretty intense here. So this kind of forms a protection for them. And apart from that, as you all know, seagrasses have a multiple function to play, and they are valuable nursery habitat for many vertebrates and invertebrates. They sequester carbon and they help in sediment stabilization. So all these benefits encourage us more, and also to transplant seagrass in different parts of the project. So we were planning about transplantation of seagrass, and what happened was, I'll explain that in detail actually in the following slides. Before going into that, I would like to show you the morphology of seagrass. This is how seagrass looks. This is the tip of the leaf here, and the leaf blade, and there is a node and internode, and the stems extends horizontally, and this is how they spread. And they have roots at each node, and they just spread laterally. They are, as you know, they are marine flowering plants, but again in here it's mostly asexual reproduction. So if you see the distribution of seagrass in Kuwait, there are 3 locations. One is Doha in the north, and Dubai in the middle, and Noiseb in the south. You have both intertidal and subtidal seagrasses in these 3 places, but again intertidal seagrasses are dominant in the south. And also, if you see in terms of area, Dubai has got a very high cover of 3.4 kilometers square. So before we go into the actual seagrass transplantation exercise, what we did in CCP, I would like to explain about what are the other restoration efforts which has happened in the Gulf with regard to seagrass. So these two papers here are Japanese papers, basically by Katakura et al. 2008 and 2013. This is about seagrass restoration, which happened in Abu Dhabi by a method called MAT method. They used a nylon mesh and fixed it to a PVC frame and then placed it close to the seagrass bed and allowed the seagrass bed grow into the mat and planted that, placed that mat in other areas where they can establish. So another exercise was Al-Jamali et al. 2015. This is the largest seagrass translocation exercise so far in the Gulf. It is a massive exercise running to more than 7 square kilometer area or something, and it has got about 85% success rate. And this particular exercise motivated us to carry out our experiment in Sea City. This is Sea City seagrass colonization. If you see here, there are several places where the seagrass is naturally colonized between 2009 to 2014. So slowly they started spreading by itself. And this area here, which is near the island 4, has got a very high cover of seagrass around spreading up to 87,302 meters square area. This is a massive seagrass bed and a very healthy bed. And from this bed, it was slowly colonizing into other places as well. So this area was a very important donor site for us. We thought of moving seagrass from this area to other places in order to assist in the colonization process. But to our surprise, what really happened is, see, this is what the natural colonization of seagrass is in Sea City. This is how it looks. It is a naturally colonized bed. It is not a transplanted seagrass bed. So when we were about to do the transplantation, so as the project was progressing into its 5th phase, this is the 5th phase, and we were actually about to transplant the seagrass that we had planned from the island 4 area. But again, what happened is there was another seagrass bed in this area, which was impacted by the construction of the 5th phase. So our management advised us to move the seagrass to this area near the island 4. So in order to assist in the expansion of the seagrass, and there were a lot of bare patches near the island 4, where, especially sandy patches where we can transplant seagrass. So both the species were there, Alophila ovalis and Haradil urinovis. Haradil urinovis was more dominant and common. So before going to the next slide. So before going into the transplantation, what we had to do before is we had to first investigate the characteristics of the donor site and the recipient site. So what we have to do is see whether the current patterns are similar, the water quality is similar, and we measured all these parameters and we decided this is the best place to transplant. So the methodology that we adopted is based on Fonseca et al. 1998. This is called SOD method. This is a very popular method used for seagrasses like Haradil urinovis, because Haradil urinovis spreads pretty quickly. And you try to transplant them through this method. When you try to adopt other methods, there is a lot of failure. So everybody recommends this method. So, and also in Qatar, they adopted this method. So we thought this method will be much more useful for our conditions. So we fabricated a sort tray with holes like this. It's about 25 into 25 into 10 centimeter. So when we have to transplant seagrass, we have to procure them from the donor site along with the sediment. So what we have to do is we have to dig through the sediment and collect the seagrass like this and keep them in a portable swimming pool with fresh seawater to avoid stress. And then we moved it with the help of a car to the recipient site. So this is how it is. See, you have to use a 25 centimeter square sort tray, collect the seagrass with the sediment, and then transplant them in the recipient site by digging 4 holes of a similar size, and then place them gently in this hole individually, and then cover them with the sediment. By doing this slowly, they will root and establish. To do this, you need a team of 4 divers. You need dedicated and trained people to do this. because this is a very, very labor intensive exercise. You have to do it very carefully. Seagrasses are very delicate. They can die in no time. So if you delay the transplantation process, they will die. So you have to take extreme care. So you have to plan accordingly, plan the logistics, and then go ahead. So this is how we did it. See, we are the diver is sliding the sod tray into the pre-dug holes, and then slowly he's just covering the sod with the substrate. And after transplantation, this is how the sod looks. You can see in this bottom picture here. And for monitoring purpose, we laid quadrants there. We had permanent quadrants. We had tagged those quadrants. We kept them monitoring for 12 months. And this is how a transplanted sod look after a couple of months. And with the help of a 25 centimeters quadrant, we started monitoring the shoot density and the leaf length. So we transplanted around 144 meter square area of seagrass in a period of 1 month with the help of 4 divers. So this much time it needs to do a small area like this is a very pilot scale study. If you are going for a large scale transplantation, you need more people. And there'll be a lot of logistical challenges that you have to plan accordingly. So this is the results of the mean shoot density for Alophila unionevus and Alophila ovalis on the first row on top, and also the mean shoot length for both the species. What happens is the density and length, these seagrasses in Gulf especially, from summer to winter, you will see a decrease in the shoot density. Similar is the growth rate also. And then after winter, by spring, they'll start growing again, because during summer they'll start shedding the leaves. And they'll almost enter a senescent stage, and then again in spring they start growing. This is how it grows here, and there is no sexual reproduction. Lessons learned. So what we have learned from this exercise, this is again a massive exercise that we did, and from this we also learned a few things. We had a lot of failures also when we did the transplantation, because in some places the current was very strong. So there we have to put some sandbags in front in order to reduce the flow. Otherwise, the sod will get uprooted. So you have to do a thorough investigation of the hydrodynamic conditions before you plan for any seagrass transplantation or restoration experiment. So because the time and money spent in this type of exercise is enormous. And also, if you don't plan accordingly, then it will lead to a massive failure. All over the world, seagrass restoration is 50-50 only. But again, if your methodology is scientifically robust and it is a proven methodology, then if you do it in a more systematic way, then the success is really high. And you should have, being a labor-intensive project, you should have trained manpower. Without a trained manpower, it's impossible to achieve this. Also, you can adopt multiple transplantation methods instead of following one method, because if you try to target from different directions for a particular species, if you try different methods, it is possible that you can achieve a success rate. But you should see which are those appropriate methods for that particular species. Then you should try. Followed by the seagrass is the coral transplantation. So in the Arabian coral nursery and transplantation studies in Abu Dhabi recently was by Wilson and Marimuthu and Sen and Yusuf. This was the recent study of coral transplantation and coral nursery in the Arabian Gulf. And there were no other studies so far in the northern part of the Gulf. And we were planning to do this transplantation experiment. So what we first thought of this, when we went through the literature, we understood that we need to establish coral nursery. So we first established a coral nursery. So in order to establish a coral nursery, we should have a proper design. So once you establish a nursery, then when you can raise corals in that nursery, then you can slowly populate them in different parts of the city and you can transplant them. So by this way, you can enhance the value of the habitat, and also slowly a lot of other organisms will colonize. This will enhance the biodiversity as well. So we targeted one species, which is Acropora downingi, and we selected a suitable site also, which I showed in the previous slide. So before going into the transplantation experiment, so I would like to tell you more about the coral nursery. What are different types of coral nurseries available? and what type of nursery we have to select depending upon our own objective and our own conditions. Like say for example, this is a floating nursery on top left here. So this is normally very useful in terms of raising a very high density of corals. So especially when you have a mid-water nursery, you can increase the height of the nursery according to your requirements. And these nurseries are very easy to very easy to construct, and also they have to be maintained with utmost care. But on the right hand side, you see the table nurseries. These are fixed nurseries. These are mostly in shallow environments and also where there is less turbidity, and these type of nurseries are very suitable. On the left here, you see that rope nurseries and tree nurseries. These are also again similar or kind of floating type nurseries, and they can be in the mid of the water column, or even just a few meters above the ground. So it can be installed, and these are very easy also to maintain. Now you can raise a huge number of corals, especially the branching corals are ideally grown through these rope nurseries. And also tree nursery. This is also another form of nursery which is very popular in America and also in other parts of the world. These are small units, and several such units can be constructed using PVC pipes and steel rods, and then you can hang the Acropora corals and any other branching coral and raise them. And apart from all these techniques, there is another technique known as biorock. This is a very popular technique, again developed by some German scientists. What happens is they use an anode and cathode attached to one of the cathode is attached to the metal frame, and there is an anode, and also they attach coral fragments to this metal frame, so that due to the electrolysis process, what happens is it forms calcium carbonate, and it allows the coral to attach and grow very quickly. Corals grow very quickly in this method. So this is also one type of method what people follow, and it is very useful to establish a nursery this way, and then take subculture from this nursery and transplant them in other places. So we looked into the donor sites. These two donor sites were there, which are about 8 kilometers from the project. These are patch trees, basically. On the donor sites, we collected those Acropora branches which were damaged by boat anchoring. We never damaged any living colony. We were only collecting those colonies which were damaged by the boat anchoring. And those colonies, from those colonies, we procured our nubbins. So these two sites here are the donor sites. So this is the Acropora downingi that we targeted. And this is the picture of nursery and the recipient site. Nursery and recipient site are pretty close to each other because it was easy for us to logistically handle them and maintain them and transplant corals from the nursery to the donor site. There were 2 donor sites, 2 nursery sites, and 2 recipient sites. In order to raise the coral nubbin, it's called a nubbin, the fragment of coral which you raise in the nursery is called a nubbin. In order to raise a nubbin in the nursery, you need to have a proper substrate. So here we did an experiment. We mixed steel slag powder. Now, steel slag is nothing but a waste product of the steel industry. And this is very rich in calcium oxides and calcium carbonate. It is pretty alkaline. So we mixed it with the marine grade cement in the ratio of 1:3, and then we use small PVC rings to cast out small button-like base like this with 4 holes. See, this is how they are. These are the slag discs. On this disc only, we transplanted we attached the fragments of the Acropora. These are the fragments that we obtained from the donor site. These were damaged by the boat anchoring. This Acropora were cut using electrical pliers, 1 to 3 centimeters size, and they were attached using super glue on these small cement slag discs. And this is the cable nursery that we designed, and this will float in the middle of the water column around 3 to 4 meters depth, and from the ground it is around, from the bottom of the seeds around 1 or 1.5 meters. So we can, if you want to lower it down, we can always adjust it. Just tied on all the 4 legs are tied with the marine grade rope. So these are the nubbins after placement in the nursery. You see here, after attaching them to the disc, they were placed in the nursery, and they were raised for almost a year in this nursery. And you can see the progress of the growth. Slowly they started growing. But again, if you see in the rest of the world, the growth rate is pretty fast for Acropora. Acropora grows more than 10 or 15 centimeters per year. Here it grows in Gulf around 10 centimeters per year. But again, in Kuwait conditions, it's even more worse. Because of the harsh conditions here, they grow a bit slowly. There was an impact in the growth. This was because our nursery site was shallow. We had a lot of biofouling, and because we do a weekly maintenance and we also monitor the growth on a monthly basis. But again, the maintenance, even though it was intensive, there was very intensive fouling happening on the substrate. If you see here, a lot of fouls are attached, and barnacles was a big menace. Barnacles were creating a lot of trouble for us. So that kind of competition is that when there is more competition, corals will have to invest more energy in establishing. That is why they were not able to grow that fast. But still, they were healthy, and they started growing more slowly day by day, and they reached almost this size. What you see here, approximately around 4 centimeter in with IC. So then slowly we started thinking of the transplantation. So then we also experimented on another nursery type. This is called rope nursery. Before going to transplantation, we also try to raise them through rope nurseries, because here they can grow big faster than the conventional nursery what we had, which was shown in the earlier slide. Because they have to invest less energy. They are just tied from a rope, and this is a small fish line from the rope. This fish line is attached to the rope, and they are just hanging from the rope. So when you attach a fragment, branching coral or non-branching coral, they will grow quickly because there is very less energy they have to spend in attaching, and there is not many fowlers attaching on a nylon fish line. So this way also it was easy to grow them. But again, we had some issues in the site, and due to some problems in the site, many of them had died. And there was a thermal plant nearby, and that thermal plant was releasing some effluents, and that created some mortality in this nursery. So we had to literally remove that nursery from that site. So this is what I'm saying. See, some of these things what I learned is just by trial and error, because you have to be very careful in the site and also the method that you're going to adopt for the nursery. This rope nursery is very effective, and also you can grow non-branching corals like Orites, Favia. Those type of corals will also be can be raised to the rope nursery. So before we transplant, so we have to select the recipient site, and you have to clean the site. You have to designate a location for each and every nubbin that you're going to transplant. So you have to clean that area, chisel it, and remove all the algae. And then you have to use an epoxy to attach the nubbin to the recipient site. So when you attach with the epoxy, then you have to look into the marine grade epoxy. Those type of epoxy will help, because the curing time should be very, very less. If the curing time is more, then it's very difficult to transplant, because you cannot be holding one nubbin for more than 45 minutes underwater. Once you attach, within few seconds, it has to set. So what we did is we did an experiment there also. We tried to use an additive, an additive, which is normally used in construction purpose. And we mixed that additive with the marine cement. This is actually a waterproofing additive, and it sets very quickly. When we mix with marine cement, the curing time was relatively low. Less than 30 seconds it set. So what we did is we tried experimenting with that type of glue, and we transplanted the corals in that recipient site at 1 minute interval from each and every nubbin. So then once you transplant them like this way, so there'll be enough space for them to spread, these being branching corals. And also we have to monitor their growth over a period of time. What we did is we monitor the growth. In case of the nursery corals, we did the monitoring for the growth by cell skeletal extension. But in case of the transplanted corals, we had to measure the geometric mean diameter by measuring the maximum the lateral length and also the perpendicular length. And then there is a formula which you can apply and take a square root to get the geometric mean diameter. This way, your data will be comparable to the experiments done in the rest of the world. So this is a survival mortality chart. So this is the first batch survival rate in terms of nursery, and also the transplants, and also this is the second graph showing the survival and detachment rate. We had in terms of first batch survival after transplantation, 43% survival rate was there. Second batch, more than 89%, because we had improved the method of attachment. We have used a different adhesive, which I mentioned earlier, the marine grade cement and the additive that we used. So that way we were able to increase the survival rate, because there was very less detachment, and whatever we attached, around 89% of them survived. So this is a transplanted Acropora downingi, which is around 3 months old after transplantation. This is 1 year after transplantation. This is about 2 years after transplantation, and we managed to publish this research in an open access journal named Cogent, which is under Taylor and Francis. And this is the coral garden after 3 years. Now it is an established population. Probably they'll be spawning now, because since I left the project, I didn't have much access to the project to do any monitoring. So, but to my knowledge, they are growing well, and slowly they are establishing a population there. So lessons learned again. So when you are trying to do a coral restoration or transportation, whatever it is, don't go only for the branching corals. You should also try non-branching corals. There should be a mixture of species, because the reason being is branching corals, they'll grow fast. They'll give you very good results. But when there is a bleaching event, they will die in no time. They are very, very fragile. Non-branching corals are very slow growing, but they are very, very resilient. It's better to go for both branching and non-branching type of corals. And also select deeper nursery sites. In our case, we didn't have a choice. We have to live with what we have. And some places where there is more deeper sites are available, you have to look into the light penetration and everything, and then you can go for a deeper site. That way, you can minimize the impact of biofouling, and the growth rate will be better. And also try different nursery types. Don't just go for only one method. If you have different types of nurseries, then you can have different type of species growing in different type of nurseries, and also the you'll have more. You'll have a regular harvest of corals from that nursery, and you can have more nubbins for transplantation, which is available at any time for you. So, and also you should also understand the zooxanthellae composition. When you try to procure coral fragments from the nubbins from the donor site, don't go only from one site. Try to go for different sites. There should be a mixture in the zooxanthellae composition. Otherwise, they may not survive on the longer run. So you have to look into the genetic pool also. There should be some genetic diversity there. So also, if you have the funding available, then if you have the money, then you can go for ex situ coral culture, doing culture in the laboratory. So when you have a different type of culture system, like land-based culture system and also in situ, then it is good to have more number of nubbins available for any transportation exercise, because some of the restoration exercises, if you are targeting a bigger area, you have to adopt different methods. Just by following one method, you won't be able to achieve the target. So I would like to acknowledge some of my colleagues and some of my mentors, my gurus, who have been very instrumental and very supportive in connecting the studies. And without them, it is impossible to achieve this. We had established 3 different habitats, but which is growing and which is also performing very well. And there is a very strong connectivity between these habitats. I'm very happy that I'm able to do that in a man-made ecosystem. And without the help of these people, it is impossible. I would like to thank all of them. And these are the references which I've used for this presentation. And if you are looking for seagrass restoration and coral restoration, these are the 2 websites that you have to look into. And also there is a very recent paper by Dr. Lisa, published in PLOS One on the coral restoration, which is a very interesting review paper, which all of you should read if you're targeting for any coral restoration project. So all this will give you more information related to coral restoration, and also this will give more information related to seagrass restoration. and you will have a lot of case studies where you can go through and understand, and from that you can design your experiment. Thank you for listening. I'm very, very happy to share what I have been able to achieve so far with all of you. Thank you so much. This is my email ID and my mobile WhatsApp number. You can just make a note of it, and any queries, I'll be more than happy to answer.
Thank you so much, sir.
Yeah.
Hello.
Yes, ma'am.
Yeah.
Thank you.
Yeah, thank you so much, sir. It was a very interesting subject, and it was very nice also. Thanks for
Thank you.
sharing your experience and new things. Thank you very much. It was really interesting. Okay. Dear participants, it's open to all. If you want to ask question, you can ask. You can unmute your hello. Yeah.
Hello. Yeah.
Participant, you can unmute your mic and you can ask the question. Some students are there. If you want to ask in Tamil also, you can ask. Answer. Sir will answer the answer for your questions.
Yes, no problem. Tamil கூட answer பண்ணலாம், ஒன்றும் பிரச்சனை இல்லை.
ஆமா, தமிழ்ல ஆன்சர் பண்ணுவாங்க சார். It is very interesting. ரொம்ப அழகா explain. Yeah, Arun, please ask your question.
Hello.
Yeah, Arun.
Yes, Arun. Hi.
Yes. Yes, sir. Sir, your presentation was very much informative and very useful, sir. Actually, I think it was a very challenging. It might be a very challenging thing. I have one simple doubt, sir. I mean, simple doubt or some kind of trial. I would like to ask. That is, have you? I mean, if can we try any genetic engineering method to improve the salt tolerance of this Avicennia marina, sir? And there are also some regulatory issues will be there, but I'm just asking, because if genetically engineered plant we can imply means it is very easy to maintain and it can survive for a long time and compared to the native one. In that aspect, I'm asking, sir.
It's possible, Arun. You know, such kind of experiment has never been done so far in the Gulf, and nobody has even thought in those lines. But if there is any good example elsewhere, we can really adopt that.
Okay.
It is not that is impossible. It is possible. So basically, you have to do it and see how it performs. Because Kuwait conditions is totally different from the rest of the Gulf. Some of the plants which will thrive in other parts of the Gulf will not thrive here because it's a very, very harsh environment. We felt raising mangroves is like growing mangroves in the moon. It was almost impossible. So somehow we succeeded. Thank you so much. Thank you.
Fine. Thank you, Arun.
Thank you.
Anybody? Yeah, thank you.
Sir, this is Subbaraman, sir.
Yes, sir.
Hello? Yeah, yes, sir.
Yes, sir.
Sir,
Yes, sir, I
in
can
the Arabian Sea, in Arabian Sea, some of the organizations transplanted corals from Lakshadweep to Gujarat and from Gulf of Mannar to Gujarat. So this is a completely habitat different. What do you think of it, sir?
Actually, Subram, it's a good question. You know, this is a very interesting experiment. The relocation experiment from Lakshadweep to Gulf of Kutch. I think one of the experiments failed. One from Gulf of Mannar to Gulf of Kutch has succeeded. Basically, these are two different environments because Gulf of Kutch is being a high latitude reef. It is highly susceptible to sedimentation. Even though Gulf of Mannar is also having a similar type of sedimentation pattern, but Gulf of Kutch is totally different. The tidal amplitude is different, so everything is different there. But again, you know, before you do this type of exercise, it's worth looking into the zooxanthellae composition, whether they can really thrive in those conditions or not. See, some corals are able to thrive in a particular environment. It's not because they are very strong. It's because of their zooxanthellae. The most important factor is the zooxanthellae. So when we don't understand that part of the coral biology properly, then doing an experiment like this is a bit risky. But I wouldn't criticize it as a successful experiment, and they have already published it also. The corals are growing well. Hope it will grow well because the acropora population in Gulf of Kutch is already extinct, so they've kind of introduced them now. So we will see how it goes. But this is what it is. it is a very big risk to do this kind of exercise. And also it's very contradictory also. You cannot, you try to publish this kind of data set, you will be subject to a lot of criticism. You have to justify that. I hope I have answered your question.
Yeah, yes, sir.
Sir, I have a question.
Yes, yes.
Hello. Sir, I have a question.
Yes.
Transplanting coral. Someone is, please mute your mic. Hello. Yeah. Hello, sir.
Yeah.
I have a question. Transplanting coral is a very tedious one. Okay. Is there any particular part to transfer the coral, or we can take any part of the coral?
See, you can just take any small part of the coral, because they will eventually encrust, establish, and then they start growing. That is how they grow. If it's a branching coral or it's a non-branching coral, same matter. You take a small fragment, even a 1 centimeter square size fragment can grow.
Okay.
It's enough.
How long it take to acclimatize and it will start growing, sir?
It depends. See, madam, it depends on the conditions of the nursery site, and so the smaller fragments might acclimatize quite quickly. The bigger fragments might take a little bit of time also. It depends on the conditions, how it is in the nursery area. And it will vary from species to species also.
Okay. Is there any criteria? Is there any essential to grow in the this one coral? Is there basic needs for the corals?
The basic need is the good water quality and good circulation. You should have a very good water quality. So if you want to establish a nursery, or if you want to do transplantation, you should understand the characteristics of both the site, the donor site and the recipient site. Both should have very similar water quality parameters. Otherwise, you will not succeed. It's very difficult because these are very sensitive animals.
Okay. Okay. Thank you, sir.
Welcome.
Participants, any questions? Participants, you can ask your questions. Students, coral, seagrass பத்தி சார்கிட்ட எதுவும் questions இருந்தா கேட்கலாம். Hello.
Yes. Hello. Yes. Hello.
Hello.
Yes. Yes, please. Yes, I can hear you. Please tell me.
Dr. M. Vairamuthu sir raised your hand. You can ask your question. Dr. Vairamuthu? Is there Dr. Vairamuthu? Hello? During your presentation, Dr. Vairamuthu raised his hand. So now you can ask your question. Yeah. Hello? You unmute பண்ணிட்டு நீங்க क्वेश्चन கேட்கலாம். ஹோஸ்ட் வந்து உங்க எல்லாரையும் அன்மியூட் பண்ணிருக்காங்க. நீங்க கேட்கணும்னா அன்மியூட் பண்ணிட்டு क्वेश्चंस கேளுங்க. Miss Malati, hello. Malati கேளுங்க, அன்மியூட் பண்ணிட்டீங்க. You can ask your question. Malati? Hello? Okay. There is a chat. Okay.
Okay.
That's all? That's all? Is there any questions? Someone is trying to ask the question, but they couldn't, it seems.
Yes. I could hear properly.
Yeah.
So everybody is clear, so no questions, sir.
Yeah. I think it's clear for everyone. Hello? Yeah, okay. Thank you so much, sir. I think,
Thank you, madam. Thank you for the opportunity.
yeah.
It's a great pleasure.
Yeah. Thank you, sir. Let me call upon. Hello? Shall we come? Subbu? Yeah, Subbu is there?
Ma'am.
Yeah, Subbu.
Yes,
I request Mr. S. Subraman to deliver your vote of thanks. Subbu, you can start. There is some.
Yeah, thank you, ma'am.
You just make Malathi to mute.
Thank you, ma'am. Good afternoon.
1 minute. Subbu, wait. Wait, Subbu. Some participant, Malathi. Yeah. Wait, Subbu. I'll mute it. Then you can speak. What is mic? Yeah, Subbu, you can start, Subbu.
Yeah, good afternoon to all. It's here, ma'am. Hearing?
Yeah, can hear.
Yeah, good afternoon to all. It's my privilege to propose a vote of thanks. On behalf of the Department of Zoology, I thank our Honorable Secretary and respected Principal for giving this opportunity to organize this webinar. On behalf of our college and management, my sincere thanks to our respected resource person, Mr. Nityananda. We are really enlightened. The habitat enhancement effort with concrete evidence in the three man-made marine ecosystem and really inspired the various techniques used for developing these ecosystems. It's really one of its kind, sir. Thank you very much, sir, for your presentation. My sincere thanks to our HOD, Dr. Bhanumathi, for her down-to-earth work in this webinar. Thank you, ma'am. And also my thanks to Dr. J. Nagarajan and R. Nagarajan for their support and coordination. I thank our colleagues, admin and superintendent, Mr. Sarwan sir, and other office staff for their technical support. I extend my big thanks to Dr. Adavan, Satyabama University, and Dr. Pungodi ma'am, ICSC coordinator, Kamraj College, and Mr. Madan, research scholar, for their timely help and support. Finally, I must mention our deep sense of appreciation to all the participants and my dear students in this event. Once again, my sincere thanks to everyone. Thank you so much.
Thank you, Subbu, for your wonderful word of thanks. Finally, hello. Yeah, we have come to an end. We know that when we protect our ocean, we are protecting our future. Quote by Bill Gates. With this note, I conclude this webinar. Once again, I thank you all for the participation in today's webinar. We hope to see you again next time. Thank you, one and all.
Thank you so much, sir.
Thank you so much.
Yeah, thank you, one and all.
Thank you. Bye-bye.
My heartfelt thanks to Sir Nityanandan, sir. உடனே நாங்க சொன்ன உடனே
Thank you.
ஒரு ஷார்ட் பீரியட் தான் இது. நம்மளால வந்து ரொம்ப கொஞ்சம் டைம் கொடுத்திருந்தா we could have done it very great manner. But ஷார்ட் பீரியட்லயும் நீங்க அக்செப்ட் பண்ணிட்டு
எனக்கு ஒரு ஷார்ட் டைம் தான் இருந்தது. பட்
ஆமா, ஒரு 2-3 டேஸ்ல ஒரு வெபினார் ஆர்கனைஸ் பண்றது வந்து it's hats off to you, sir. That is this is possible only because of you. I am very grateful to you always. Okay.
Thanks, madam. Thanks for inviting me.
Yeah, sir.
Thanks for inviting me.
Most welcome. Whenever you come to India, you please do visit our college and motivate our students,
definitely, I will.
Zoology students, young students,
Definitely.
to reach a big height.
Definitely.
Definitely. We welcome you, sir.
Sure, madam. Sure.
Okay.
All the best.
Yeah, thank you so
All
much.
the best. Thank you so much. Thank you,
Aran
Mr. Somu.
Jayan,
Thank you so much.
பேசுங்க.
Thank you,
நித்யானந்தன்
sir.
சார்ட்ட பேசுங்க. ஜெயன், ஆரன் ஒரு 5 minutes பேசுங்க சார் கிட்ட. ஹலோ ஆரன்.
Hello.
ஹலோ.
Sir,
Hello.
Nityanandam sir.
Yes, sir.
Sir, நான்
சொல்லுங்க sir.
மாஸ்டர் டைவர் தான் சுப்ரமன்.
Okay, சொல்லுங்க சார், சொல்லுங்க.
கல்ஃப் ஆஃப் கட்சில நான் தான் சார் ஒர்க் பண்ணேன்.
நீங்க தான் ஒர்க் பண்ணீங்களா? சரி சரி.
சுப்ரமன், கௌதம், பிரேம் ஜோதி 3 பேரும் சேர்ந்து ஒர்க் பண்ணோம்.
சரி சரி. கௌதம் எல்லாம் சேர்ந்து எப்படி இருக்கு கோரலாங்க இப்ப?
இப்போ கொஞ்சம் பரவாயில்ல சார். இப்போ அக்ரபுரா லட்சத்தீவுல இருந்து டிரான்ஸ்பிளான்ட் பண்ணது நான் தான்.
அப்படியா? சரி 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 சரி. அது ஃபெயிலியர் ஆயிடுச்சுன்னு கேள்விப்பட்டால கொஞ்சம்.
இல்ல, ஃபர்ஸ்ட் எஃபர்ட் வந்து ஒரு 6 monthsல ப்ளீச்சிங் ஆயிடுச்சு. லோக்கல் கோரலே ரொம்ப ப்ளீச்சிங் ஆயிடுச்சு. மாஸ் ப்ளீச்சிங் நடந்துச்சு. அப்போ செகண்ட் தேர்ட் நாங்க அட்டெம்ப்ட் பண்ணல.
சரி. இது ரொம்ப ரிஸ்க் சுப்பு.
ஆமா சார். நாங்க கிட்டத்தட்ட 1 year ட்ரை பண்ணி சக்சஸ்ஃபுல்லா டிரான்ஸ்பிளான்ட் பண்ணிட்டோம். அது வந்து மோர் தென் 8 months வந்து சக்சஸ்ஃபுல்லா இருந்துச்சு சார். அந்த ஆகஸ்ட் மந்த் பீக் ப்ளீச்சிங் டைம்னா அந்த சமயத்துல தான் ப்ளீச்சிங் ஆச்சு.
ஓகே ஓகே. இதுக்குதான் நான் சொல்றது, அந்த ஜூசான்தலை காம்போசிஷன் வந்து இன்னுமே சரியா படிக்கல இந்தியால. சோ இப்பதான் படிச்சுட்டு வராங்க. சோ அதுதான் ரொம்ப இம்பார்ட்டன்ட். அத ப்ராப்பரா புரிஞ்சுக்கணும் நாம டிரான்ஸ்பிளான்ட் பண்றதுக்கு முன்னாடி.
சரி.
கல்ஃப் ஆஃப் மன்னார்ல இருந்து போனதும் நல்லா இருக்கு இல்லீங்களா இப்ப?
ஆமா சார், கல்ஃப் ஆஃப் மன்னார் நல்லா இருக்கு. இல்ல, கல்ஃப் ஆஃப் மன்னார் சேம் தான். அவங்க வந்து அக்ரபுரா ஃபார்மோசா பண்ணிருந்தாங்க.
ஓகே.
நாங்க ஹிம்லிஸ் மட்டும் பண்ணிருந்தோம். அவங்க அக்ரபுரா ஃபார்மோசா போக ஒரு 13 ஸ்பீசிஸ் பண்ணிருக்கோம்.
சரி 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 சரி. ஓகே.
எக்ஸெப்ட் ஹிம்லிஸ்.
ஓகே ஓகே. குட், வெரி குட்.
நாங்க ஹிம்லிஸ் வந்து அங்க ஏற்கனவே ஸ்கெலிட்டன் ரெக்கார்ட் பண்ணிருக்காங்க. சோ அதனால ஹிம்லிஸ் மட்டும் நாங்க ஃபோக்கஸ் பண்ணிருக்கோம்.
இது பப்ளிஷ் ஆச்சுங்களா உங்க ஸ்டடி இல்லையா?
ஆமா சார். டேரிங் டு ரீஸ்டோர் அப்படிங்கிற புக் என்னோட பேர்ல பப்ளிஷ் ஆயிருக்கு.
சரி, சந்தோஷம். வெரி குட். Congratulations.
Thank you, sir. Now I will send the copy, sir.
Please.
Sure.
நித்யானந்தன் சார், நான் உங்களை பார்த்திருக்க சான்ஸ் இருக்கு. நீங்க இசட்ல அந்த டைம்ல இருக்கும்போது நான் சிபால இருந்தேன் சார். நம்ம முட்டுக்காடு சிபா சென்ட்ரல்.
நான் டாக்டர் வெங்கட்ராமன் கிட்ட தான் இருந்தேன். அவரோட ப்ராஜெக்ட்ல தான் இருந்தேன்.
அடுத்து ரகுநாதன் சொல்லிட்டு ஒருத்தன் இருந்தார்ல. ரகுராமன் சாரி ரகு.
நேஷனல் பயோடைவர்சிட்டி அத்தாரிட்டில
சார்.
நேஷனல் பயோடைவர்சிட்டி அத்தாரிட்டில இருக்காரு.
ஓகே. அவரை மீட் பண்றதுக்கு நான் வந்திருக்கேன். அப்புறம் உங்க சாரை பார்க்கும்போது வந்திருக்கேன். சோ ஐ மைட் பி சீன் யூ. எனக்கு சரியா ஞாபகம்.
ஓகே ஓகே. எனக்கு உங்க ஃபேஸ் கொஞ்சம் ஃபேமிலியரா தான் இருக்கு. எங்கயோ பார்த்த மாதிரி தான்.
சிபால தான் நான் SRF இருந்தேன். அங்கதான் PhD டாக்டர் ஆசாத்.
சரி சரி.
டாக்டர் ஆசாத். டாக்டர் ஆசாத் தெரிஞ்சிருக்கும் உங்களுக்கு.
அவர் குவைட்ல தான இருக்காரு.
இப்ப குவைட்ல தான இருக்காரு. ஆமா.
என்னோட கலீக். நான் அவரோட கலீக்னு சொன்னோம். அவர் ரொம்ப சீனியர் ஆக்சுவலா.
சாரோட ஸ்டூடண்ட் சார் நானு.
அப்படியா? சரி சரி.
சாரோட ஸ்டூடண்ட். ப்ராஜெக்ட்ல சார் கிட்ட தான் இருந்தேன்.
ஓகே ஓகே ஓகே.
எல்லார்கிட்டயும் சொல்லிப்பேன், கடவுள் தான் எனக்கு சார் தான் கடவுள். அவ்வளவு டவுன் டு எர்த். He taught me a lot. நிறைய விஷயங்கள் அவர்கிட்ட இருந்தா கத்துக்கிட்டது. Really such a wonderful person. இப்போ நான் எத்திராஜ்ல படிச்சேன். Yeah, very humorous guy. Very good gentleman. நல்லா நான் நினைச்சேன் சார்
15 years இங்க 15 years
சார் 2003ல போயிட்டாங்க சார் அங்க.
ஆமா
3 or 4ல அங்க வந்தாங்க.
எக்ஸலென்ட் ஜாப்.
நான் 3 to 4ல வந்து சப்மிட் பண்ணேன். நான் அதை கேட்கணும்னு நினைச்சேன் உங்ககிட்ட அப்பவே. ஆசாத் சார் இருக்காங்க அப்படின்ட்டு.
சொல்றேன் நானு.
சொல்லுங்க. சரி, உங்க SRFன்னு சொல்லுங்க, டக்குன்னு புரிஞ்சுப்பாரு.
கண்டிப்பா.
ஆல்வேஸ் நம்ம ஒரு SRF தான் மைண்ட்ல டக்குன்னு வரும். இப்ப வந்திருந்தாங்க சார் ஒரு இன்டர்நேஷனல் கான்ஃபரன்ஸ்க்கு எத்திராஜ் காலேஜ்க்கு லாஸ்ட் இயர்.
எத்திராஜ் காலேஜ்க்கு வந்தாங்க. சரி.
ஆமா, எத்திராஜ் காலேஜ்க்கு வந்திருந்தாங்க.
சார் ரெகுலரா நிறைய கான்ஃபரன்ஸ் அட்டெண்ட் பண்ணுவாங்க.
வருவாங்க. அதான் உங்களை மாதிரி நான் ஈஸி அக்செப்டபிள் பர்சன், ஈஸி அப்ரோச்சபிள் மேன். நல்லா
ரொம்ப ஹியூமரஸான ஆளு கூட.
ஆமா, சரி. கன்வே மை விஷஸ் டு ஹிம் சார். If possible, please share his email ID so that I can.
Yes. நான் உங்களுக்கு அனுப்புறேன். No problem.
Okay. Thank you so much, sir. Yeah.
My pleasure, madam. Thanks a lot.
We also
keep in touch. Thanks a lot.
Yeah.
Thanks for inviting me.
Yeah, thank you.
Take care. Stay safe. Bye.
Yeah, bye, sir. Thank you, sir.
Bye. Bye, Subbu. Bye.
Bye, sir.
Participant, you can fill the feedback form. It is in the chat box. Dear participants, you can fill up your feedback form. Feedback form fill பண்ணாதான் உங்களுக்கு certificate வரும். So all of you kindly fill up your feedback form. fill பண்ணிட்டு உடனே போய் மெயில் செக் பண்ணுங்க. உங்களுக்கு வந்துட்டு உங்களோட certificate ரெடியா இருக்கும். இமிடியேட்டா வந்துரும். Dear participants, இன்னும் ஒரு 5-10 minutes தான் இருக்கும். 5 minutes தான் இருக்கும் ஃபீட்பேக் லிங்க். நீங்க போயிட்டு பண்ணிடுங்க. Dear participants, you can go and fill up your feedback form so that you can get the e-certificate immediately through your registered email ID. You can go and fill up your feedback form. After that, you cannot get the feedback link. So do it immediately. Subbu, Jayan, Aran.
Yes, ma'am.
Khanu, Jayan. Hello.
Yes, ma'am.
Okay.
மேடம். ஆரன் பேசுறேன் மேடம்.
பேசுங்க, பேசிக்கலாம்ல. சீஃப் கெஸ்ட் கிட்ட சொன்னேன்ல. சும்மா 2 வார்த்தை பேச வேண்டியது தானே. ஹலோ
மேடம். ஹலோ.
ஹலோ
மேடம், நல்லபடியா பண்ணி முடிச்சிட்டீங்க. Thank you.
All of your support and cooperation. இது வந்து ஒரு நல்ல இனிஷியேட்டிவ் நினைக்கிறேன். நமக்கு நல்ல ரெஸ்பான்ஸ் வந்திருக்கு இ சர்டிபிகேட்டுக்கு வந்து குவிஸுக்கும்.
ஓகே மேம்.
குவிஸுக்கு வந்து புரியல எது
சாட் பண்றது நம்ம
எல்லாருக்கும் தான் இருக்கும். இருங்க, நான் அதை இது பண்ணிடுறேன். சாட் பண்றது ஹலோ சுப்பு கேக்குதா?
மேடம் கேக்குது மேடம்.
பேசுங்க. நான் இப்போ ஹோஸ்ட் ஒன்லி தான் போட்டிருக்கேன்.
சரி மேம்.
நம்ம 4 பேருக்கு மட்டும் தான் கேட்கும். நம்ம ஹோஸ்டா யார் இருக்காங்களோ அவங்க மட்டும் கேட்கலாம்.
மேடம், இப்ப நிறைய பேர் உள்ளக்க ஜாயின் பண்றாங்க.
இல்ல, நான்
சுரேஷ் பாலன்
வரல, அக்செப்ட் பண்ணல. இப்ப அக்செப்ட் பண்ணலையா?
சரி மேடம். சரி.
நான் அக்செப்ட் பண்ணல. நான் இப்ப அந்த வோட் ஆஃப் தேங்க்ஸ்க்கு அப்புறம் வந்தவங்கள யாரையும் அக்செப்ட் பண்ணல.
ஓகே மேம். ஓகே
மேம். ஜெயன் சார் அக்செப்ட் பண்ணுங்க.
ஜெயன்யா
எனக்கு ஃபோன் பண்றாரு.
என்னமா? ஆமா, வெளியே இருக்காரு. பண்ணிட்டேன். ஆனா சுப்பு,
மேம்.
ஹலோ. நமக்கு ஷார்ட் மினிட்ல ஓகேயான ரெஸ்பான்ஸ். நமக்கு டைம் இல்ல, அது ஒண்ணு.
குயிக்கா பண்ணி கொடுத்தாரு.
பிரேக் ஆகுது மேடம்.
பெரிய எனக்கு தெரியும் எந்த அளவுக்கு கஷ்டம். அதான் சரி, டிரைவரா எனக்கு தெரியும் எந்த அளவுக்கு கஷ்டம்ட்டு. ஏன்னா நாங்க ஒரு 100 ஃபிராக்மெண்ட்ஸ் டிரான்ஸ்பிளான்ட் பண்றதுக்கே ரொம்ப கஷ்டப்பட்டோம் பண்ணியிருக்கோம். பட் இந்த அளவுக்கு அவங்க எஃபர்ட் கொடுத்துருக்காங்கன்னா பெரிய அச்சீவ்மெண்ட் தான். அது உண்மையிலே பெரிய விஷயம்.
நல்லா பண்ணியிருந்தாரு, பெரிய ஒர்க். நினைக்கும் போதே கஷ்டமா இருக்கு, அது எப்படி பிராக்டிகலுக்கு கொண்டு வராங்களோ தெரியல.
ஆமா, அதுவும் 3 ஈகோ சிஸ்டமும் பண்ணிருக்காங்க. பண்ணும்போது அது எல்லாமே ரொம்ப நைட் ஒர்க் தான், உங்களுக்கு சொல்ல முடியாது. ரொம்ப நல்ல ஒர்க்.
இட்ஸ் வெரி டீடியஸ் ஒர்க் கூட.
ஆமா, நிறைய இன்ஃபர்மேஷனும் ஷேர் பண்ணியிருந்தாரு. அது பார்க்கும்போது எந்த அளவுக்கு அவங்க ஹார்ட் ஒர்க் பண்ணிருக்காங்க, எந்த அளவுக்கு நாங்க கம்மியா பண்ணிருக்கோங்கிறது தெரியுது.
இல்ல, அப்ப இருந்த ஃபெசிலிட்டிஸ் வேற, இப்ப இருக்கிறது வேற இல்ல.
அது ஒண்ணு இருக்கு. நான் சொல்றது 10 இயர்ஸ் பேக் நாங்க ஒர்க் பண்ணியிருந்தது. இந்தியா லெவல்ல அந்த இது கொஞ்சம் கம்மி தான். பட் ஆனா இந்த அளவுக்கு அவங்க எஃபர்ட் எடுத்து பண்ணிருக்கிறது, நியூ ஐலாண்ட் கிரியேட் பண்ணி அதை டெவலப் பண்ணி கொண்டு வரதுங்கிறது சேலஞ்சபிள்.
ஓகே. எனக்கு டவுட் இருந்தது, எங்க அந்த ஆசாத் சார் அங்கதான் இருக்காரு. எனக்கு ஒரு டவுட்ல தான் இந்த இன்ஸ்டிடியூட்டா வேற இன்ஸ்டிடியூட்டான்னு டவுட் இருந்து. குவைட்ல இருக்காருன்னு தெரியும், அவர் எடிட்டோரியல் போர்ட்ல எல்லாம் இருக்காரு. அதனால தான் ஒரு டவுட் கேட்டேன் நான் அவர்கிட்ட. ஓகே, இருக்காங்க போல பார்ட்டிசிபென்ட்ஸ்.
ஃபீட்பேக் வந்து அப்படியே குயிக் பண்ணலாம்.
சார் ஃபீட்பேக் ரன் ஆகிட்டு இருக்கு.
சரி ஓகே,
ரன் ஆகட்டும். இங்க 35 தான் வந்திருக்கு.
டோட்டல் பார்ட்டிசிபென்ட் எத்தனை பேர் வந்திருக்காங்க?
அரௌண்ட் 48 தானே இருந்தது.
அவ்வளவுதானா?
ஆமா, அதுல 48ல ஓகே 15நாலும் உள்ள நம்ம ஸ்டாஃப்ஸே வந்து எத்தனை பேர் இருந்தது? லைக் பானு மேம், நீங்க, ஜெயன் சார், ஆரன் சார், நான், அப்படியே வந்து அப்புறம் உங்க ஆஃபீஸ் ஸ்டாஃப்ஸே ஒரு 3 ஐடி இருந்தது. ஸோ இதுலயே ஒரு 8 to 9 ஐடி. இப்போ நமக்கு 40 தான் வரணும், இப்போ 38 வந்திருக்கு. இன்னும் இருக்காங்க. நம்ம செல்வகுமார் ஃபில் பண்ணிட்டாரு, செல்வகுமார் உள்ளதான் இருக்காரு.
நம்ம ஸ்டூடெண்டே கிட்டத்தட்ட 100க்கு மேல வந்து குடிக்குதுன்னு அவன் ஃப்ராட் பையன்.
இந்த வெளியில இருந்து வராங்க ஃப்ரெண்ட்ஸ்.
Hi Banu, this is Sharmi.
Sharmi.
ஐயோ, ஏன் கட் பண்ணாம இருக்கீங்கயா? நான் மெசேஜ் போட்டேன், நீங்க பார்க்கல.
ஓகே ஷர்மி, how are you?
Yeah, fine.
அப்புறம்
actually well, sorry, personal லைன்ல அப்புறமா வரட்டுமா?
ஓகே ஷர்மி,
இப்பதான் ஜாயின் பண்ணேன்.
ஓகே,
நீங்க லைன் கட் பண்ணாம பேசிட்டே இருக்கீங்க. ஜூம்ல, யூடியூப்லயும் இதுக்கு அப்படியே வருது லைவ்ல.
அப்படியா? சரி ஷர்மி, நீ வா, நீ இப்போ வா. வெல்கம். இவங்க காகிதம் இல்ல ப்ரொஃபசர், மை ஃப்ரெண்ட் ஷர்மி. ஹலோ, ஆசாத் சாரோட கலீக் தான் இப்ப பேசினது.
ஓ ஓகே ஃபைன். லாங் பேக் we met, right? ரொம்ப வருஷம் ஆயிடுச்சு சார் பார்த்து.
நம்ம இதுல பார்த்தோம்ல இப்போ பிப்ரவரியில.
எஸ் இவங்க ரிட்டையர்மென்ட் வந்திருந்தாங்கல.
நம்ம ஒரு கான்ஃபரன்ஸ்ல பார்த்தோம்ல பிப்ரவரியில.
ஆமா.
ஓகே. Hello.
Okay, Banu, I'll catch you later.
Yeah. You come, Sharmi. You come in. I'll let you in.
Yeah, இதுல ரூம்ல கனெக்ட் ஆயிருக்கேன்ப்பா. Through that
இங்க உள்ள இருக்கீங்க. ஆமா. Right. Sorry.
GSM இருக்கும்.
GS. Okay. Fine. Okay.
வேற டிரைவர் இருக்கா பாருங்க.
Okay, Sharmi.
Okay,
hello.
Bye.
Thanks for joining us.
Thank you.
Yeah. Bye.
ஆரன் சார் கொஞ்சம் இதுல வாங்க, வீடியோ ஆன் பண்ணுங்க.
Kindly change the host to Anthony Solomon. Power problem in office.
ஆனந்த் சார் கொஞ்சம் வீடியோ ஆன் பண்ணுங்க.
ஹலோ
ஹலோ
சுப்ரமணியன் சார், நீங்க பேசுறது பேசுங்க சார்.
சார் வீடியோ ஆன் பண்ணுங்க சார்.
வீடியோ இருக்கு சார், பேசுங்க நீங்க.
இல்ல பின்னால பாய் ஒண்ணு போட்டிருக்கீங்க ஸ்கிரீன்ல, நல்லா இருக்கு, அதான் கேட்டேன்.
கட் பண்ணி விட்டேன். வீட்டுல உட்கார முடியல, வெக்கையா இருந்துச்சு, அது வெளியில வந்துட்டாங்க. ஹலோ
சார்,
பேக்ரவுண்ட் கண்டுபிடிச்சீங்க சார் என் பேக்ரவுண்ட.
அதான் அனுப்பினல, இட்ஸ் லுக்கிங் குட்னு. இட்ஸ் லுக்கிங் குட் அப்படின்னு சொல்லி அனுப்பினல, அது வந்து நான் பாய தான் சொன்னேன்.
அப்படியா? நான் பார்க்கல. நான் வந்து நீங்க அனுப்பின மெசேஜ் பண்ணீங்களோ?
ஆமா ஆமா.
சரவணன் சார். ஹலோ சரவணன் சார்,
சொல்லுங்க மேடம்.
முடிச்சிடலாமா சார்? எக்ஸிட் ஆகிறோம், டைம் ஆயிடுச்சு 3:51. ஹலோ
எஸ் மேம்.
ஹலோ
மேம், எக்ஸிட் ஆயிடலாம்.
ஓகே ஓகே, தேங்க்யூ.
ரொம்ப
ஓகே மேடம். நீங்க ட்ரான்ஸ்ஃபர் ஆயிட்டு
சரவணன், ரொம்ப நன்றி.
தேங்க்யூ சார். தேங்க்யூ சார்.
சரவணன் சார்,
தேங்க்யூ சார்.
ஓகே, தேங்க்யூ சார். தேங்க்யூ
ஓகே மேடம். ஓகே மேடம். தேங்க்யூ மேடம்.
ஓகே, தேங்க்யூ so much. Jayan, Aran, Subbu, thank you so much for your cooperation. Thank you. Bye. Yeah, bye.